In this video, I'm going to go over all the biochemical reactions that we use to help us identify specifically gram-negative bacilli or gram-negative rod bacteria. They are quite a few biochemical reactions. We normally go over these in 2 weeks, 2 separate times of setting up various tubes, and then we spend another lab reading and analyzing all the tubes. So I'm going to make it as one big video going over all the different ways we set up our gram-negative biochem reaction tubes. I'm also going to go over a couple tubes. There are biochemical reaction tubes, but they are 2 tubes that we use for separating out or identifying gram-positive cocci that are catalase negative. So I recommend having both. If you're going to watch this video and fill out the worksheet as we go along, having both biochemical reaction worksheets up, ready, printed, however you're going to work on it. Have both of them. I'm mostly just the way I'm going to set them up, going to kind of flip back and forth a little bit just on how I inoculated the tubes. So have both of them ready. The worksheets are set up that at the very top, the A, B, C, D, it's how you set up the tubes. Are you using loops? Are you using needles? Are you stabbing all the way to the bottom? Are you just zigzagging the slant? So it's just how you set them up. And I'm going to start there. So I'm going to go over how we set up all of the various tubes for the gram-negative bacteria, and then how we set up 2 of the tubes for a gram-positive bacteria. So just have the worksheets ready. Again, you can pause anywhere in here, anywhere in there. You can use the lab exercises that are on Blackboard as well to help you fill out the worksheets. You can use the PowerPoints to help you fill out the worksheets. But I do have a couple videos mixed in that I took in lab of me actually setting these up. So our starting, all of these biochemical tests are really the majority of them are to help sort out gram-negative rod bacteria, because most gram-negative bacteria are bacilli or rod-shaped. And so just doing a gram stain is not enough to help narrow them down. So all of these biochemical tests, whether it's for gram-negative or for positive, is because we add a substrate into the media, and we're going to look to see if it makes a particular product. If it does, there's going to be some indicator in there, just like on the plates, but it's now going to be in tube form. We're going to look for a color change. That indicator is going to tell us whether this product can be made or not, because not all organisms can break down certain substrates and make products, and we're kind of just asking a lot of yes/no questions. Now, this is kind of a flow chart. It's not every test that we're setting up, but it really is a yes/no flow chart. Can it ferment or lactose? And we can see that on a McConkey plate, but you can do it in a tube as well. And if you ask enough yes/no questions, is it modal or not? Does it make hydrogen sulfide or not? Can it break down gelatin, yes or no? Like, if you ask enough yes/no questions, you can eventually ID out the organism. And that's what we're doing. We're setting up various types of tests to look to see if the bacteria can do various things or not. So the 4 bacteria that we work with in this lab, because they don't all show us positives on everything, we have to look for differences between them. That's the whole point of this, is looking for differences. And of these 4 organisms, all of them are, one of the 4 will show us a positive reaction. The 5 tubes that we're going to set up for our gram-negative bacilli or gram-negative rod bacteria are these 5 tubes, and I'm going to go in order. So I know these are not the order on your biochem reaction tubes, but this is the order I videotape and actually set them up. So it's the order I'm going to discuss them. So the first one is the lysine iron, LIA, the triple sugar iron, the TSI. Those are on your biochem reaction B sheet. The MIO, the citrate, and the urea are all on your biochem reaction A sheet. And then I do have an oxidase test that I don't set up in a tube, because it's a non-tube test that we can set up. That's also for your biochem reaction A. So starting with the first, we have the lysine iron auger. It starts off as a nice purple color. We pick up, using the needle, we pick up a good chunk of organism. And I'm like, several colonies if you need to. You stab all the way to the bottom of the tube twice. You stab all the way down, pull it up, stab all the way down, pull it up again, and then you take that needle and just zigzag up the slant. So you're getting lots of bacteria down in the butt region of the tube, but you're also getting some of that bacteria on the slant region of the tube. And best results is if we have a tight cap. Everything else is loose caps, but this one actually gives us a little bit better results if we have a tight cap. The bacteria will do their color change that they're going to in 24 hours. With a tight cap, they'll have enough oxygen in there. The next tube we're setting up is the triple sugar iron auger. And again, you can pause me anywhere in here. We're also using a needle for this one. We're just picking up a colony from a plate of bacteria. We're using that needle stab to the bottom of the tube, and then we zigzag with the needle going up the slant, depositing bacteria in both the butt region and on the slant region, and we have a loose cap. The third tube we're going to set up, this is from the Biochem Reaction A sheet, is the Mio tube. It's Mio because it's actually testing 3 different things: motility, indole, and ornithine breakdown. And we're still going to use a needle. We're going to pick up a colony of bacteria, and we're just going to do a stab about halfway, 2/3 of the way down, and then pull it up. There's no zigzagging because there's no slant. We're just stabbing it 1/2 to 2/3 of the way down. The other 2 tubes that were set up are both from the Biochem Reaction A worksheet. It's the citrate and the urea, and we set both of them up the exact same way. We're going to use a loop instead of a needle. We're going to pick up a colony of bacteria from a plate, and we're going to start at the bottom of the slant and then zigzag up the slant, just depositing bacteria in the slant region, and we do that with both the citrate and the urea. I was going to say I mixed my order up. This is just to show the Mio tube is showing or can test for several different things. It will test for motility, it will test for indole production and the ornithine decarboxylase. This tube, although it does test for motility, it's not the only way that you can test for motility because sometimes it's not the most reliable. Other ways that we can test for motility, we can stain the bacteria. If you see flagella, they're motile. You could look them underneath the microscope and look to see if they're swimming around, or you can use something like the Mio tube, the motility auger, to see if they move away from the stab mark. Now, the last test, and this actually kind of gives you your results as well, and I put the results down in the table itself, is we're looking to see if your bacteria has a chemical called cytochrome C. Now, how we end up doing this is we take just a little snippet of filter paper, we put a drop of the enzyme oxidase on it so you can see the wet spot from that oxidase, and we take a loop and we just rub some of your bacteria right on that little wet spot on the filter paper. If the bacteria has the chemical cytochrome C, it gets broken down, and it actually shows us as a purple color. And so sometimes we can see the purple color. Not of the 4 bacteria that we are working with, only one of them is positive for oxidase, the Pseudomonas. And otherwise everything else just looks like a little bit of bacteria smeared on a white piece of filter paper. So on the Biochem Reaction A sheet in the table down below, again, you're just putting a drop of, for how you do it, it's just a drop of oxidase on the filter paper, rubbing some of the bacteria colony with a loop onto that wet filter paper, and you're looking for the color change. Down below, under the results, I already list, substrate product. We're just looking for a particular chemical called cytochrome C. The indicator is the enzyme, and a positive reaction is that you're gonna see a purple pigment in the bacteria. The bacteria is gonna show up as purple. Now, after 24 hours, we're going to have some test results. We're going to be able to see some of these different tubes. We're going to be able to see their different reactions. We're going to see the color changes that they make.
So we're going to set up our biochem tubes, inoculating all 5 of our biochem tubes. Again, our 5 biochem tubes are to help sort out gram-negative rod bacteria. So we're going to use the needles that you guys didn't get a chance to use this semester. And I am going to be inoculating all of my tubes today in the video using E. coli bacteria. So now that my needle is cooled off, I'm going to pick up an isolated colony of bacteria. And the first tube I'm going to inoculate is the Lysine iron auger. It's going to start off as a purple color. This one, they want you to have a good amount of organism. I can see it on my needle. We're going to stab to the bottom twice. I'm going to stab all the way down, lift it up, stab all the way down in another location, and then I'm still going to zigzag on the slant, still getting some bacteria. This one also seems to work best if it has a tight cap. All of the other ones get a loose cap. I'm going to incinerate my loop before I pick up some more bacteria for the next biochem tube. The next biochem tube we're going to set up is going to be our triple sugar iron auger. Again, I'm going to pick up another colony of bacteria. I can see it on my loop. This one gets one stab to the bottom of the tube and a zigzag right up the slant. And it gets a loose cap, so I always tighten it and then do a quarter turn back. The next tube we're going to set up is our MIO. It tests 3 different things. It tests for motility, it tests for ornithine production, and it tests for indole production. Get that tube ready. Again, I'm going to pick up another colony of bacteria. So luckily I've got a lot of bacteria on my plate. And this just gets one stab about halfway, 2/3 of the way down. One stab, and then that's it. Loose caps, do a quarter turn back. Then we're going to get rid of our needle and switch to the loop for the other 2 tubes. The first tube that we're going to set up is called the citrate. It's a nice and a pretty green color, but it can get even prettier the next day. I'm going to pick up some bacteria from my plate, and this just gets a zigzag on the slant. So I'm using the loop, no stabbing whatsoever, and I'm just zigzagging up the slant. A loose cap. Incinerate the loop. Cool off. And then our very last tube that we're going to set up is called the urea auger. It kind of has a peach color to it to start off with. Just loosening my cap to get it ready. I'm going to pick up some more bacteria from another colony. And this is also just a zigzag on the slant, starting at the bottom and just zigzagging up to the top. And this tube also gets a loose cap. The only cap that we have as a tight cap is that first tube, the Lysine iron auger. Incinerate my loop, and we're done inoculating. Then we just have to incubate and wait 24 hours.
All right, let's take a look at results. So we incubated those initial tubes, everything all testing for gram-negative rod bacteria. And again, there's a lot of gram-negative rod bacteria. Now, out of the 4 bacteria that we are working with, E. coli, Klebsiella, Proteus, and Pseudomonas, the reason I put this slide up there is one of them's not like the other. And so because Pseudomonas is a strictly aerobic bacteria, it has results that indicate that it's a strictly aerobic bacteria. So when I go over the positive and negative results of these 4 bacteria, you'll notice on some of them, Pseudomonas gives some strictly aerobic bacteria results. These bacteria, these other 3 that are in the Enterobacteriaceae group, they're facultatively anaerobic, which means they can grow in both aerobic and anaerobic conditions. Now, the first tube we had set up, it was purple to start off with. It's called the Lysine iron auger. Now, it's testing one tube to look to see if your bacteria can break down lysine. And so when we're filling in the biochem reaction B worksheet, we're looking at the LIA tube. So it's the 3rd row on the table down, and the substrate is lysine. So we're looking to see if your bacteria can break down lysine. Now, if it can, the product, if it can break down lysine, is just called lysine byproducts. It's not a specifically named product, it's just lysine byproducts. Now, the indicator to tell us if lysine was broken down, it's a pH indicator is written on here, called bromocresol purple. Now, positive reaction. It can vary because there's two different ways that you can break down lysine. You can either decarboxylate it or you can deaminate it. Lysine is an amino acid, and on one side of the amino acid it has an amino group, and on the other side it has a carboxyl group. And so some bacteria can break down lysine by removing the carboxyl group, and other bacteria remove the amino group. That's what the D-amino for deaminate means, or decarboxylate is removing those different groups. So it just depends on which way it breaks down the lysine, which side of the lysine it breaks down. And depending on which side it breaks down, does actually give us a color change difference. Now, if it can decarboxylate it, it only does it in the anaerobic part of the tube, so down in the butt. And what we're looking for is if the bacteria, or if the tube has a purple butt. If it does, if the butt region, because we always indicate what's happening in the slant and what's happening in the butt, if the butt region of the tube is purple, it means it's positive for decarboxylation. If the slant region is red, that means the bacteria is able to break down the lysine, but it does it through deamination. So you're looking either for a red slant or a purple butt to know if it can break down lysine. However, if you have a purple slant over a yellow butt, as indicated by these two or these two, it means it can't do anything. It cannot break down lysine whatsoever. Not all bacteria can. So we're looking to see if it can break down lysine, and then if it can, does it have a red slant for deamination, or does it have a purple butt for decarboxylation? So in that last column on your worksheet for a positive reaction, the two choices are a purple butt is positive for decarboxylation, but the other positive reaction is a red slant, which is positive for deamination. So there's two positive reactions. A purple butt is positive for decarboxylation. A red slant is positive for deamination. Now, the next tube we set up was the triple sugar iron, and it started off as a nice orange color when we inoculated it 24 hours ago. And based on its name, it does have 3 sugars in it. It has glucose, it has lactose, and sucrose. I bolded and made 2 of them bigger than the other because glucose does not have a lot of glucose in it. So it's 0.1%, whereas there's a lot of lactose and sucrose in there. But it does have 3 sugars. We're looking to see if your bacteria, if it's able to ferment or break down lactose or sucrose or both. Now, anytime you ferment a sugar, you do get an acid. And so we are going to have a pH indicator. But my note, there is, other than it does have 3 sugars in it based on its name, it does have one other substrate in it, and it's peptone, which is a protein. So on your worksheet for TSI, there are 2 rows. I get this. There's a TSI fermentation and there's a TSI H2S. H2S is coming up. We don't care about that row right now. We're going to fill out the TSI fermentation row. So the substrate on your triple sugar iron is glucose, there is some there, lactose and sucrose, and peptone. So you really have 4 substrates in here. We're really focusing on the lactose or the sucrose and not the glucose of the 3 sugars though. So 4 substrate, glucose, lactose, sucrose, and peptone. If the bacteria can break it down, depending on whether it breaks down sugar or depending on whether it breaks down the peptone, there are 2 possible products. The first product is fermented sugar. It's not a cool specifically named product. The other product that could be made if the bacteria breaks down peptone is just metabolized peptone, which means broken down peptone. So you got 2 choices for product, fermented sugar or metabolized peptones. Now, if you ferment a sugar, you're going to get an acid, and if you metabolize peptone, you're going to get a base or an alkaline product. And so our indicator is another pH indicator. It's phenol red. Now, depending on whether the bacteria can ferment the sugar or whether it can metabolize the peptone, depends on where it's happening in the tube. So sometimes some of this happens up in the slant, sometimes some of this happens down in the butt region of the tube, and so we're going to indicate what's happening in both regions. We're always going to write it down as what's happening in the slant, what's happening in the butt. Now, if we see acid being produced, which is going to show up as yellow, we're going to indicate acid with an A. So we're going to say if there's any acid happening in the slant, and if there's any acid happening in the butt, giving it an A if you see yellow for acid. If we see any breakdown of peptone, that's going to produce an alkaline product, and because we can't use A, we're going to use K for alkaline, and so we're going to say whether something's happening in the slant or the butt. If we see any red, we indicate that with a K. If it looks just as orange as it did when we first set it up, we put an NC for no change. And if we notice any gas being formed, like carbon dioxide gas, usually we would see that if there's air bubbles or the media cracks apart, we just indicate that with a G. There's really no breakdown of lactose or sucrose happening or peptone. It's just sometimes we do see gas being produced. So, some possible positive reactions. There are multiple positive reactions, write small. So for that last box on your table for a positive reaction for TSI fermentation, one possible positive reaction is A over A. Now, what that will look like, because these are the 4 bacteria we set up, A over A, this just indicates that we see some gas. A over A means there's acid in the slant, acid in the butt, and we see that in both E. coli and Klebsiella. We've got acid in the slant, acid in the butt. Now, K over K I don't have a picture of. That would indicate there is all red in the tube. K over no change I do have, though. It's our Pseudomonas. It breaks down peptone, but it does nothing with the sugars whatsoever. And so we do have metabolized peptone up in the slant, and we have nothing happening, no change, still orange, happening down in the butt. Now this is where that Pseudomonas being strictly aerobic comes into play. The only place Pseudomonas is going to do anything is up in the slant, and it breaks down peptone only. Now, another tube that we have, Proteus. It is K over A. However, Proteus is positive for our next thing, the TSI tests, and so we can see the red in the slant. The yellow is a little hard to see. We know there is yellow, but that black is covering it. Out of another possible reaction, these 4 bacteria aren't showing it. If you have K over A, K is for alkaline for red, A is for acid, which is the yellow in the tube. Again, we're always recording what's happening in the slant, what's happening in the butt. Now, possible positive reactions. I know I kind of skipped ahead and I may have lost you. I apologize. One possible positive reaction is A over A. Another possible positive reaction, K over A. That's what this is showing, this tube, but harder to see. Another possible positive reaction, K over K. That would be an all red tube. These tubes shown here don't show that, but K over K would be an all red tube. And K over NC, or K over no change. Now, Mike, which we see with our Pseudomonas. So these are all possible positive reactions. Lots of things happening. Lots of different colors happening. Now, the triple sugar iron TSI does another test as well. It tests for hydrogen sulfide formation, so this is that next row on the worksheet, the H2S. Our substrate is thiosulfate. Our product is hydrogen sulfide gas. Our indicator is ferrous sulfate. And a positive reaction, we would see black in the tube. And we see a lot of black in the tube. So out of the 4 bacteria we work with, Proteus is the only one that can break down thiosulfate and produce the hydrogen sulfide gas, but we see a black precipitate. So a positive reaction, black precipitate. So we've got our substrate, our product, our indicator, and our positive reaction. So we're done with the TSI tube. So now we're going to go on to the Mio tube. This is on the other worksheet. We'll come back to bile esculent sodium chloride broth later because they're for gram-positive bacteria. But the Mio tube tests for 3 things, so it gets 3 rows on the worksheet. Now, the first thing that it gets, I filled it in for you. It's testing for motility. Now, we did a half to 2/3 of the way stab down with the needle, And what we're looking for is if the bacteria are motile, they will swim away from the stab mark. And so you're going to see a cloudiness around the stab mark. If it was not motile, it would stay right in the stab, it would not move. So I already filled in the table. We're just looking for a little turbidity, a little cloudiness around the stab mark. If there's only growth in the stab mark, it's non-motile. However, we're going to look at the ornithine breakdown. And there are several things happening in this tube when it sits in that incubator for 24 hours. Now, just so that I make sure you guys get your table filled in, this is for the ornithine row of the MIO. Your substrate is ornithine. And we want to know if your bacteria can break it down. If it can, the product is called putrescine. And so we're looking for putrescine, spelled right here. Now, it is an alkaline product, so we want a pH indicator. And so our indicator, it's a pH indicator, our indicator is bromocresol purple. So the substrate is ornithine, the product is putrescine, and our indicator is bromocresol purple. Now what we're looking for in the tube, in the bottom of the tube, we are looking for purple. We want a purple butt. So if you have a purple butt in the bottom of the tube, that is positive for ornithine breakdown. Now, my note, some things that are happening. This tube started off as purple. So how do you know it did anything at all? So there is some glucose in there as well. So when we inoculate the tubes, they start off as purple, but we also know most bacteria can break down glucose. They can ferment glucose, and they're going to produce an acid, and the bottom of the tube turns yellow in the incubator. We want to know, when the glucose runs out, can they then break down ornithine? And if they can, they produce an alkaline product, so we go back to purple. So there's color changes happening in the incubator. It starts off as purple, turns yellow, but then we want to know if it can turn back to purple. So a purple butt is positive for ornithine breakdown. We do run into one issue though. So we're looking to see if it did this color change, but when we pull out the tube 24 hours later, what happens if it didn't do anything? Because Pseudomonas, which is a strictly aerobic bacteria, won't do anything down in the butt of a tube because there's no oxygen. And yet the bottom of the tube is purple. So generally we will write that down as a positive reaction. You have purple butt, it's positive. However, you just kind of have to note at the back of your mind that a purple butt is positive for ornithine, but it could mean, if you have a strictly aerobic bacteria, that it's actually negative or no change, that it hasn't done anything. Now, the other test, the Indole test for the MIO, and we do it last for a reason, is because we have to add something to it. Now, for the Indole test, our substrate is tryptophan, and we want to know if our bacteria can break down tryptophan. If it can, the product is indole. So it's called an indole test because we're testing for the product indole. So substrate is tryptophan, product is indole, and our indicator is called indole reagent. It's a reagent that tells us if there's indole present. Now, a positive reaction is we're going to add indole reagent. So we have to physically add something to it. It's not already added. A positive reaction is that indole reagent, they say it's called cherry red. It's more hot pink. And I'm like, but officially it's cherry red. So we're going to look to see if the indole that we add turns hot pink or cherry red. So a positive reaction, cherry red ring at the top, because that's the indole. I'm like, but it looks hot pink. Oh, here is my tryptophan, indole, add indole reagent, and we're looking for the cherry red ring at the top. Now, 2 out of our 4 bacteria are positive for making indole. They can break down tryptophan. Now, we got just a couple more tubes left, and I'm like, the first one is citrate out of our last 2. This is also on the Biochem reaction A sheet. This was a slant. It started off purple, and we want to know. So here's I'm writing everything down. Citrate is the substrate. The product is just alkaline products. It's not a specifically named product that you have to know. The indicator, it's another pH indicator called bromothiamyl blue. And a positive reaction is if you have any blue in the tube. And it's a really pretty royal blue color. So it started off as just all green. It's the one we used just a loop. We just did a zigzag on the slant, but we're going to look to see if there's any blue. So a positive reaction is blue. And then our last tube is the urea tube. This is another one we just zigzagged on the slant. Our substrate is urea. The product is ammonia. Now, carbon dioxide is formed, but we're not testing for it. So we're looking to see if ammonia is produced. It's an alkaline product, and so our indicator is another pH indicator called phenol red. So substrate urea, product ammonia, indicator phenol red. Now, a positive reaction is if there is hot pink. It's super pretty. Hot pink anywhere in the tube. Even if you have just a little bit of hot pink in the tube. We call it a weak reaction, but there's hot pink being formed, which means ammonia is formed. So we're just looking for hot pink. Now, this is just to show some of the test results. So we did set up each of our 4 bacteria. I'm just going to kind of go over some of the test results, some of the unique things. So for the LIA, and again, if you want to be following along with the tables you just filled out, the LIA, we're looking for if the butt of the tube is purple, and it is, so it's positive for decarboxylation. The TSI tube we record as A over A for yellow over yellow. For the E. coli, it is positive for motility. I zoomed in on it. We had a stab mark, but you can really see all that cloudiness around the stab mark. It is positive for ornithine breakdown because it's got a purple butt, and it's positive for indole productions. We would add indole to the tube, and there's that hot pink indole fluid on the top there. Citrate is negative, we don't see any blue, and urea is negative, we don't see any hot pink whatsoever. This is just to show what the original colors looked like. You can see how the TSI started off orange, turned yellow. The Pseudomonas that we tested, although the bottom of the tube for the LAA is purple, we could record that as a positive breakdown of the decarboxylation. However, I also know, or the breakdown of lysine, sorry, Mike, but I also know that this is a non-modal, or it is a strictly aerobic bacteria. It's not going to do anything in the tube, so it stays purple. It's negative. The TSI, this is the one we record what's happening in the slant and in the butt. The TSI tube does show red up in the slant. I try to zoom in. Red up in the slant, but the bottom is still orange, so it's no change. So K for red for alkaline over no change. Motility, it's positive. Because it's that strictly aerobic bacteria, you don't see cloudiness around the stab mark because there's no oxygen down in there, but we do see a complete covering and growth. It's hard to take a picture of that on here across the whole surface. It's indole negative. It would not show the cherry red ring. Ornithine, although it's purple, it doesn't do anything because it's a strictly aerobic bacteria. Citrate, here's that nice pretty blue up in the slant region. Definitely positive for the urea, because it's got that nice hot pink in there. Proteus, for the LAA, we have red up in the slant, indicating the positive deamination. In the TSI, we have red in the slant, yellow in the butt, so K over A, but it's again hard to see the yellow because this is positive for hydrogen sulfide, that black precipitate. So again, this is what the tube originally looked like, is orange, doesn't look like that anymore. For the MIO, it's positive for motility. It does have the cloudiness around the stab mark. It's negative for indole production, doesn't show the cherry red ring, and it does have a purple butt, so it's positive for the ornithine breakdown. No blue, negative for citrate, definitely hot pink, so it's definitely positive for urea. And then the fourth bacteria that we worked with, the LAA, it's got a purple butt, so it's positive for decarboxylation. A over A for yellow over yellow, or acid over acid. The MIO, there's no movement around the stab mark. It does have pink. I was trying to show that. There is pink for indole production, but the bottom of the tube does not turn purple. So it uses the glucose, turns yellow, and then it can't break down ornithine. So the butt stays yellow. So it's negative for ornithine. It does have a nice blue up in the slant for positive for citrate, and definitely hot pink for the urea. We had a couple more biochem tubes to help us sort out some gram-positive cocci bacteria. So in lab, we identified some of your nose bacteria or some of your skin swabs as gram-positive cocci. We did a catalase test. They all showed up as catalase positive. However, if they would have shown up as catalase negative, no bubbles, that narrows down to streptococcus or enterococcus. But that's still two big genera of bacteria. So these next two tubes are to help separate out which is which. So if you had a catalase negative bacteria, you would set up these two tubes and it would tell us whether it's a streptococcus or an enterococcus. Now, we are going to work with an enterococcus bacteria. So you're going to see what positive results are for an enterococcus, which means if these tests would show up as negative, it would be a streptococcus bacteria. So it's setting up two tubes. So these are the last two tubes on your biochem reaction B sheet, the bile esculent and the sodium chloride broth. We're going to use loops for both of them. We're going to pick up some enterococcus bacteria, and on the bile esculent tube, we are just zigzagging up the slant. That's it. Just picking up some bacteria with the loop, zigzagging up the slant. That's how you inoculate your bile esculent tube. For the sodium chloride broth, we are picking up with our loop another colony of enterococcus bacteria, because we know it's enterococcus. You're going to put it in some broth, just some sodium chloride broth, swirl it around a little bit to try to ensure some of the bacteria get off of your loop and into the sodium chloride broth, and that's it. So it's just zigzag up the loop for the bile esculent, adds a colony of bacteria into that broth culture itself. Then we're going to incubate it for 24 hours.
Our last tubes that we're going to set up are going to help us narrow down enterococcus from streptococcus bacteria. Again, they're both gram-positive, they are both cocci shaped, and they're catalase negative. So this is a test to help narrow down that it's an enterococcus and not a streptococcus. So I do have a plate of enterococcus bacteria, and I'm going to inoculate two tubes, a bile esculent and a sodium chloride broth. So I'm going to pick up some bacteria, so I know it's an enterococcus, and our test results will show us enterococcus positive results. So our first tube, the bile esculent tube, I pick up some bacteria with my loop, and I am just zigzagging up the slant. Gets a loose cap. I'm going to incinerate. Pick up some more enterococcus bacteria. And this is a sodium chloride broth. That's a 6.5% sodium chloride broth. That's a lot of salt. I'm just adding some bacteria to the broth. I'm swirl it around to make sure some of the bacteria get off of the loop. It's not going to look very cloudy because what we're looking for is to see if our bacteria, our enterococcus, can grow in that high salt content. If it can, in the incubator for 24 hours, we're going to see a nice cloudy look to it. So right now it doesn't look like a lot of bacteria, but enterococcus should show us some bacteria.
It's 24 hours later. Again, we're using these two tubes to tell us that it's enterococcus, and so we're going to see positive enterococcus bacteria. If these tests would show up as negative, we know it would be a streptococcus bacteria. Now, for filling in your table for the bile esculent row on your Biochem Reaction B worksheet, your substrate is esculent. The product is glucose and esculetin. The indicator is ferrous citrate. And a positive reaction is black in the tube. So substrate esculent, product glucose and esculetin, indicator ferrous citrate, and a positive reaction is a black color in the tube. Now, this is to show a negative reaction. This is what the color of the tube looked like before, kind of a yellowish, smoky, grayish yellow, lots of black. If it was a streptococcus bacteria, it would look like this. It would look just like it started off day one. For the sodium chloride broth, we're looking for turbidity or cloudiness. Enterococcus bacteria can grow in a high salt content, like staph can. And so we're looking for growth. Ours is never this cloudy, but any cloudiness indicates the bacteria has been growing and is okay at that high salt concentration. Negative, it would be just as clear as it started off day one. Now, enterococcus bacteria, they're going to be positive for both. They're also going to be resistant to both antibiotics, optichin and bacitracin, because we also use this, and if we're trying to know what streptococcus bacteria, if we think it is a strep, we would test the sensitivities to these two antibiotics. Enterococcus is resistant to both, which means it would grow right up to those paper discs that are soaked with these two antibiotics. So we set these up in lab, and here's our enterococcus bacteria. This to show that tube turned super black. Negative bile esculent is still a yellowish color. I couldn't get, when I took a picture, it didn't quite look as yellow as it was supposed to, but there's no black in it whatsoever. This is to show, here's that cloudiness. It's not super, super cloudy, but it's turbid. That means this is a positive reaction. Our enterococcus would be growing and showing that turbidity. This would be some other type of streptococcus or other non-enterococcus bacteria. Wouldn't grow in that high salt content, and it would look just as clear as it did the other day. And so on your worksheet for the sodium chloride broth, there is no substrate, there is no product, there is no indicator. A positive reaction is just turbidity or cloudiness in the tube. So hopefully I answered all the questions. Again, you might have to look through some of the lab exercises. You can look at the PowerPoint itself and flip through and stop anywhere you want to fill it in as well.